Hey, Damien.
You guys. Happy New Year.
Yeah, Happy New Year. Great seeing you.
Yeah, we'll give it a minute or so. Sneha, what's coming up next week?
Next week, Dr. David Goldfarb from BCCI, BC Cancer Children Hospital, he will giving talk.
And is that part of the PALS?
It's a clinical round.
Okay. Could you just tap me an email with a description of what's happening so I can announce it at the end?
Yeah, sure. Okay.
Thanks. Okay, so because we have a very busy schedule, we're going to start now, and I know there will be some people joining us over the next few minutes. So this is our first province-wide pathology rounds for the year, and we've got a very exciting lineup here today. We have two, we say, junior members of our department who have had very important papers. Yemin Wang's paper was published in Nature Communications just a couple of months ago, and today we heard that one of the papers which Dr. Ali Bashashati will be presenting has also been accepted in Nature Communications, which is really exciting. One of the things which has been suggested is that the rounds are used to celebrate very specific accomplishments, and so this has come together well. I think that the two presentations will be exciting to the broader pathology community. So the first presentation by Dr. Yemin Wang will be about rare cancers and some phenomenally exciting work he's doing, and I'm pleased to collaborate with him on. Dr. Wang is a PhD scientist who trained at UBC. He then went to the Fred Hutch to do a postdoc, and then we were fortunately able to recruit him in 2012 to my research team and the Gynae Cancer group, where he's built up a research program where he studies rare cancers. He's a staff scientist at BC Cancer, an adjunct professor in our department. He has actually led his own grants, as well as being absolutely fundamentally important to the work I do, and in particular on the two cancers which he's going to be describing today. And so after Yemin's talk, Dr. Ali Bashashati, who was recruited to our department along with biomedical engineering, will be giving a presentation on AI-assisted cancer biomarker discovery in the digital pathology era. And Dr. Bashashati is the lead for artificial intelligence and bioinformatics research at Ovcare, and he sort of functions extremely well at this interface between computational biology, engineering, and biomedical sciences. And he's brought a tremendous edge to our research in pathology AI and how to improve the diagnosis of gynecologic cancers, but is doing similarly exciting work across other cancers as well. So we are really excited to have him present his work today. So we're going to start with Dr. Wang's presentation, and then we're going to have some time for some questions, but I'm going to have to be quite strict with time, and then we will quickly move on to Dr. Bashashati's presentation, and then we'll have time for questions after that as well. So thank you all for attending, and to Dr. Wang and Bashashati for presenting at our first province-wide pathology rounds of the year. So Yemin, would you like to grab the screen and start presenting?
Sure. Thanks, David. Can everyone see my screen well?
Yes, certainly.
Sure. Thanks, David. Hello, everyone. It's a great pleasure to have this opportunity to speaking at today's rounds. As David mentioned, I will talk about our studies in rare cancers. Why do we study rare tumors? As we know, rare tumors are very challenging to study due to the lack of fundings and resources, and therefore they are often neglected. However, those rare cancers, although they are individually very rare, and collectively they can represent 20% of human malignancies, and there's absolutely unmet clinical needs to improve our understanding of this forgotten species and to improve the patient care. In addition, from the scientific point of view, study rare cancers can be very beneficial. Unlike common cancers, which are usually heterogeneous and complicated at the genomic levels, so challenging for study, rare tumors are usually driven by a single or very limited epigenetic or genetic events, and that's very ideal for genomic discovery and for modeling the disease to understand how a normal cell acquire mutations to drive the malignant transformation. And also findings from rare cancers often are generalizable to common cancers. One famous example is a disease called retinoblastoma, and RB1 gene was found out that turned to be a causal gene for this rare cancers that cause develop tumors in young kids that impact the vision of the eyes. And RB1 was later on discovered to be ubiquitously important tumor suppressors in cell biology and also inactivated in many other cancer types. So today I'm going to talk about two rare cancers. The first one is our cytolytic cell tumors, which is a histologically very diverse mixed sex called stromal tumor. As you can see here, the morphology can vary from very differentiated to very poorly differentiated, and often they can contain heterozygous elements, and tumor can present very differently with retiform morphology. They arrive usually in the young women and girls, and they are usually associated with androgenic effects that's believed to represent some kind of a pseudo-male glidogenesis in the ovary. They run sporadically mostly, but also can occur in families, which we'll talk shortly. So when I first joined David's group in 2012, his team used next generation sequencing, and I identified hotspot mutations in one of the two enzymatic domain of DICE1 gene, which is critical for microRNA biogenesis. And these mutations occurs in both in the sporadic and familial cases. And they usually are heterozygous mutations, but the second allele tends to be lost due to either germline or additional somatic events, suggesting that the missense hotspot mutations in this enzymatic domain as predominantly expressed functional domain in cancer types with this mutation. And this study was validated by multiple groups, including additional cohorts. I had an opportunity to work with Dr. Fred Komos from Germany, and also my colleague Tony Kalisis, who has moved down to California at UC University of California. So we found that it represents roughly 60% of the sertoli cell tumors, and also occurs in the ovarian angioblastoma, which is considered as a morphological variants of sex-caused stromal tumor. We provide genomic evidence at DICE level that is indeed the case. And from this German cohort, we further, Tony and I further found that DICE1 mutation correlates with a younger age of diagnosis and also higher grade of the tumor and the presence of heterozygous elements and the morphologic different from the retiform morphology. And those high-grade tumors and the presence of heterozygous elements and retiform morphology are usually associated with poor prognosis. And highlighting this lead to understand the biology in tumors with DICE1 mutation and to identify targeted therapeutics to treat this disease. So with that, we also find that not only in these rare cancers, DICE1 mutations occurs at a lower frequency in common cancers. Particularly, we work with my colleague Yemin Chen, and we found that about 2% of endometrial cancers have these hotspot mutations, but those cancers are generally difficult to study with a high genetic mutation background. We also find that, intriguingly, these genomic mutations also extended to a whole spectrum of rare disease, which is called DICE1 syndrome. This is a rare genetic condition that first discovered by Dr. Ashley Hill at WashU, and is caused by loss of function of variants in the DICE1 gene. So loss of DICE1 germline mutations predispose the patients, usually kids and young adults, to the development of both benign and malignant cancers. This includes sertoli cell tumor, which I have just described, and also more commonly the pleuropulmonary blastoma, which are simplified as PPB, and many other rare tumors that occurs in various organs. And most recently, Dr. Felix Komos and Will folks teamed up together and utilized whole methylome analysis and discovered that this DICE1 syndrome-associated cancers, most of them are sarcomas, but regardless of anatomic sites, they share very common methylation profiles, suggesting that they likely arise from cellular origin that's from different sites that with same developmental states. And these tumor sarcomas particularly can be further divided into 3 clinically meaningful groups. The low-grade mesenchymal tumors are usually very cystic with very little mesenchymal, and usually those mesenchymal even present, they are usually hypocellular level. And occasionally you can see some rhabdomyoblastic differentiation, but at high-grade tumors, those sarcomas are predominantly mesenchymal and with higher cellularity and increased frequency of anaplasia and tumor necrosis. Also see increased frequency of rhabdomyoblastic differentiation, and molecularly targeted mutational analysis finds frequent PIP3 KRAS mutations at roughly 30% and 20% of cases respectively. They are usually mutually exclusive, suggesting a cumulative mutation may drive the development of aggressive sarcoma. And the third group called, so both low-grade and high-grade tumors, they have the epithelial lining, and underneath the epithelial lining, there's beautiful cambium-like layers, which represent undifferentiated cells that presumably has the potential to develop into various differentiated fate of the cells. And the third group, which doesn't have the epithelial lining and presence in the brain, which is called primary intracranial sarcoma, also called CNS DICE1 sarcoma. And they share most histological features as a high-grade sarcoma of the dicer, and has pure mesenchymal morphology because there's no epithelium components. And molecularly, they have even higher frequency of PIP3 and KRAS mutation, with PIP3 is mutated in over 80% of the cases. And those high grades and and brain CNS sarcoma are associated with poor outcome, and so absolutely is a need to identify the treatments for those patients. To do this, to develop therapeutic means, we need to understand what DICE mutation do to impact the cells. As we mentioned, that DICE1 is important for processing microRNA. And what it does is use two enzymatic domains to cut away the loop structure of the helping precursor microRNA and release the 5' end or 3' end to incorporate into the RISC complex to guide the target selection and subsequent gene regulation. However, with this mutation, we'll lose ability to process and cut away the loop structure from the helping precursors and systemically disable the generation of 5P microRNAs. And this will have profound impact on the gene expression. And using, we profiled a few cases of cytolytic tumors with mutants and wild-type dicers and found that many genes engaged in gonadal development are underregulated in cancers with DICE1 mutation, and particularly a gene called CYP19A1, which encodes aromatase, which is essential for converting androgen to estrogen in granulocyte cells, are significantly underexpressed in cytolytic tumors compared to granulocyte cell tumor. But intriguingly, in those cytolytic tumors with DICE mutation, you could see much lower expression compared to those without, and suggesting that the DICE1 mutation will be associated with even stronger androgenic effect in SLCT. And this is indeed supported by a study 2 years after we made this discovery by a Japanese group. So collectively, this suggests a model that a certain developmental stage of granulocyte cells, by accumulating DICE1 mutations and a global loss of 5P microRNAs, may direct the genes not only essential for differentiation, but also for oncogenic transformation that ultimately drives the pseudo-oncogenesis, pseudo-male gonadogenesis and oncogenesis in the ovary. And to test this, I have initially focused on putting DICE1 mutation into immortalized granulocyte cells from IVF patients. While there are some kind of biological effects which are quite consistent with what we observed in clinical samples, we failed to generate tumors from when we engrafting those cells back into the mice. And the lack of relevant disease models for not only SLCT, but also the entire DICE1 syndrome-associated cancers make it very risky to rely on this artificial model from immortalized granulocyte cells and to inform biology understanding, also therapeutic development. So to tackle this challenge, I developed an inducible DICE1 mutant mice that express DICE1 RNA3B mutation only in Cre-expressing tissues. And this allele functions as a low allele before Cre induction due to an altered splicing event in where we did a genome targeting. However, when we cross this strain with a fully crossed allele, which in the cells without Cre induction, it functions as a hemizygous Y-type allele, and upon Cre induction can switch off the Y-type fully fluxed allele to allow allele and activates the missense allele. So ultimately, the cells expressing after Cre were expressing hemizygous mutants. This perfectly recapitulates the genetic state of DICE1 in patients with these mutations. And together, we started crossing these two compound heterozygous two strains with multiple Cres, initially focusing on developing cytolytic cell tumor models, later on try to expand to capture the full spectrum of DICE1 syndrome disease. And we got lots of tears over years, and also have enjoyed when we have two successful models has been developed recently. So today I'm going to focus on the first model we developed using the MHR Cre, which is an anti-Müllerian hormone receptor 2 Cre driver that activates transgene Müllerian mesenchymal cells and also the pre-granulocyte cells. And in this MHR Cre-driven mice, all the females in about a year all develops para-ovarian cyst or mass, and some of them also have uterine mass. Occasionally, we see 2 lung masses in tumors, which we believe is a kind of misfiring events. And these para-ovarian cysts are usually multilobule and often very bloody. And when we open the tumor, fix the tumor and did a histological assessment, we could confirm that this cysts are of a tuber origin, and we can see very clear and expansion of mesenchymal cells in those under those epithelial lining in the cyst that Dr. Blake Jukes helped to make the diagnosis of Mullerian adenosarcoma, which is also a rare disease under the rare tumor that you can see in the DICER1's associated tumor spectrum. And so in some cases, you could see those cystic tumors completely switch to the fully solid tumors that has the morphology which fully resemble the human disease with beautiful epithelial lining and cambium-like layers and the mesenchymal expansion. And in some cases, rhabdomyoplastic differentiation can be also observed. Using immunohistochemistry, we further confirm the presence of mesenchymal cells such as segmentin and expression of desmin. And those tumors are selective for inhibin alpha expression, which suggests they are not sex-core stromal tumors. In contrast, they express beautiful WT1 in the mesenchymal component, and also ER was expressed in both epithelial and some of the mesenchymal cells, which is consistent with the diagnosis of Mullerian adenosarcoma. And using microRNA sequencing, confirm the deregulation of the 5P microRNA species, particularly NAT7 families and MIR10, MIR26 microRNAs. And whole exome sequencing and targeted sequencing also confirm there's accumulation of PIP3 and KRAS mutation, suggests that these tumors not only morphologically mimic, genetically mimics Dicer syndrome-associated tumors, but also has the molecular accumulation of additional genetic events that match the human tumors and provides reliable model studies Dicer syndrome-associated cancers. And by comparing the bulk transcriptome of this Mullerian mouse murine tumors to the oviduct from the normal mouse, we could find that this activation of MYC target pathway and HF pathway suggesting that potential therapeutic oncogenic mechanism to drive the oncogenesis, and also targeting them may be a possible approach to treat the tumor. So we are actually working on that angle. And based on this unique and the second model, which is even improved model with lineage track capabilities. So we formed 2 collaboration with colleagues in Montreal, led by Dr. Will Fox, Sid Huang, and Mark Fabian, and also colleagues in UCSD, led by Kenny Chen, and supported by DICER1 syndrome registry in Minnesota, led by Dr. Chris Ann Scouts. So we will use these models to try to address how tumors, how normal mesenchymal progenitor cells at defined stage of development that accumulates mutation and inactive microRNA biogenesis and drive cystic mesenchymal tumor development, and how accumulation of additional genetic events, particularly PIP3 KRAS mutation, drive the development of aggressive sarcoma, and ultimately use functional genomics and drug screening to identify targetable therapeutic targets, and also candidate drugs, particularly from FDA-approved drug library, to have a rapid translation to help patients at the bedside. And while slowly cooking the eggs for the DICER1 syndrome-associated cancers have been also engaged in studying several other rare cancers, particularly have spent many years working on the small cell carcinoma of the ovary hypercarcinic type, should simplify as good tumors. So this is a rare disease first described by Dr. Scowley in 1979, and the median age of diagnosis was in mid-20s. And it runs as similar as dice-associated cancers, can be either sporadic or familial cases. And unlike DICE1 tumors, these tumors are growing rapidly. They are usually poorly differentiated with some small round cells and a variable amount of rhabdoid cells and larger cells, which kind of confusing because the tumor itself called small cell. And 60% of the tumors, patients can have hypercalcemia, which is confusing too, because this tumor itself, the name of the disease is called a hypercalcemic type. But to my knowledge, this is trying to distinguish with another small cell ovarian cancer that's occurring in older patients with neuroendocrine phenotype. These tumors are usually very aggressive, 2-year survival, generally under 35%. So in 2014, several groups around the world, including Dr. David Huntsman's group, found that this is a monogenic disease driven by inactivating mutations in the gene called SMARCA4, which is the central ATPase of the Schwann-Smith chromatin remodeling complex. And this inactivating mutations cause the loss of SMARCA4 protein. And in familial cases, usually first events occur as germline events and second event somatic, that's completely losing. So it's functional, unlike DICE1, it's a classic tumor suppressor gene. And in cancers with SMARCA4 has been loss has been shown that they rely on the alternative ATPase SMARCA2 to survive, and people has the interest to develop strategies to target SMARCA2 to treat those patients, especially those small cell lung cancer patients. And surprisingly, my colleague Tony Canisius and I use various approach to check SMARCA2, especially when we found SMARCA2 is also silenced, but this is at epigenetic level. So suggesting this is a disease with completely loss of the Schwann-Smith ATPase activity. And to see whether it's a specific tool for diagnosis of SCUTs, Tony performed IHC on over 3,000 primary gynecologic tumors and found that SMARCA4 is predominantly lost in SCUTs, suggests it's highly sensitive. More intriguingly, SMARCA4 loss is also quite selective, as only a small portion of clear cell ovarian cancer has SMARCA4 loss, but those cases are usually SMARCA2 intact and also occurring older patients. And we also find that uterine sarcomas, 8% of them can have SMARCA4 loss. Some of them also have a companion SMARCA2 loss, but they also occur in older patients in general. And also a very small portion, roughly 1% of high-grade endometrioid carcinomas can have SMARCA4 loss, including one cases of dedifferentiated endometrial cancer, which can also have SMARCA2 loss. So this finding suggests that SMARCA4 loss with age of diagnosis as another factor, it can help to provide very reliable, specific, and sensitive tool for SCUT diagnosis. And with this, Tony and I has been able to reclassify 2 cell lines. One of the cell line called 4C4 was initially diagnosis as a juvenile granulocytoma, but with reanalyzing the cell line and the primary tumor that we obtained from Holland, we will be able to confirm that this is a bona fide SCUT tumors, and the cell line itself is reclassified in SCUT cell lines. And similarly, 211-2D was initially diagnosed as intermediate differentiated ovarian endometrioid carcinoma, but we found this a lesion of dedifferentiated component that lost SMARCA4 in those primary tumors, and reclassify this tumors as dedifferentiated endometrioid ovarian cancer. And Tony and others further extends finding in the initial cohort and identified dedifferentiated SMARCA4/A2 deficiency are indeed present in a significant cases of dedifferentiated endometrial cancer and undifferentiated uterine sarcomas. And those patients are generally older, but it's even but much younger, generally older in the dedifferentiated endometrial cancers, but relatively younger in the undifferentiated uterine sarcomas. And they are always associated with poor outcomes. The difference between two cancers at genomic level showing that different cancers are usually MMR deficient, but the undifferentiated uterine sarcoma with SMARCA4/A2 deficiency generally has lower mutation, which mimics SCOOT tumors. And those two deficiency has been also observed in other cancers, particularly lung cancers has been reported even before we made a SCOOT discovery in the very minor cases, but has been completely neglected. And also, since the SCOOT discovery, a lot of interest in looking to SMARCA4 deficiency by HC or sequencing, and there's a new entity called SMARCA4 deficient undifferentiated ceramic sarcoma has been made, which occurs in heavily smoker patients that usually have PIP3 mutation, and therefore harder to study as well. And SMARCA4/A2.
Yemin, sorry, we are at the half hour now, so we are going to have to move towards a rapid wrap up here to give Ali time. So sorry for interrupting you.
It will be very quick. Fairly quick now. So studying SCOOT will be very useful for understanding the spectrum of the disease, with that of, unlike the Dices syndrome associated cancer that lacks models, there are 3 SCOOT cell lines available, so that made it possible to study this cancer. So working with colleagues at UNC, Buddy Weissman and Martin Hurst, Greg Maureen at UBC, we performed epigenetic transcriptomic and proteomic analysis on SCOOT cell lines re-expressing SMARCA4 and observed massively increased chromatin accessibility in enhancer regions, and that usually activate the genes engage in cell differentiation. Particularly, we observed increased epithelial cell markers suggest a potential origin of cells of the SCOOT that may come from cells has a normal differentiation capacity to differentiate into epithelial components, lineage. Also perform the drug functional genomic proteomic analysis, try to identify multiple target therapeutic candidates, and initially focusing on targeting epigenome. I lead that work and find EZH2 DAC inhibitors along or in combination are quite effective in preclinical models, and working with Jeff Chen in TGN, we also find palatinib, which is a tyrosine kinase inhibitors, are effective in a couple of preclinical models. And later used with my CHR grants that I have been able to identify several metabolic reprogramming events in SCOOT tumors that can be targeted at different modules. And more intriguingly, although these SCOOT tumors are generally quiescent with very low mutation burdens, there are limited clinical evidence showing that they are actually response to immune modulation therapies. More importantly, so several findings from those study SCOOTs has been found to be applicable to more general disease, such as lung cancers with SMARCA4/A2 deficiency, and sometimes only SMARCA4 deficient tumors also respond in a similar way. So suggests that SCOOT is an excellent model for making discovery and extending to pan cancers with the similar genetic mutations. And however, given the rareness of these tumors, only limited trials can be run in SCOOTs. With so many options, it becomes a question which shot on goal we should take for clinical trials. With that, I have been formed a collaboration with the support of CISO CAN grants with Dr. Sid Huang at McGill to test different combinations, use both cell line derived xenograft models and connection of PDX models we have recently assembled. And so in summary, studying rare cancers not only, and we presented perfect examples that not only improves the diagnosis, but also can provide excellent models to understand the biology of general important tumor suppressor genes to deconvolute the disease development continuum. And also identify novel therapeutics not only impact these rare cancers, but also extended to other common disease. With that, I'll concludes by thanks everyone engaged in the project, particularly mentor Dr. David Huntsman over the past decades, and all the funding agencies. Thanks, everyone.
Thank you. So maybe we'll have time for one question now, and maybe some more questions at the end. If anyone would like to ask a question for Yemin, please. Okay, so what we'll maybe do is have questions at the end then. So thank you. And so without further ado, I'd like Dr. Bashashati to grab the screen. And Ali, we look forward to hearing about your work, and in particular, congratulations again for the great news on the most recent paper. Well, Ali, we can't hear you. You're listed as muted.
So I think, is it good now?
Yes, absolutely.
Okay.
Thank you.
Okay, great. Great. Thank you very much. And Happy New Year to everybody. And hope you had a great holiday break, and happy to be presenting today. So I'll try to go a bit faster than I planned so that we actually, I can finish on time. But what I'm going to talk today about is on basically artificial intelligence-based, cancer biomarker discovery, and with the focus on digital pathology. So, so I think most of you now know about, I'm probably used ChatGPT. There has been lots of breakthrough AI advances in non-medical fields that you can actually, ChatGPT is one example, Bard AI from Google, Llama from Meta. So basically they do the same thing. And even with these models and platforms, you can now chat with images, ask questions about images. You can create new images and, chat with voice as well. So within the oncology space, so I actually did a query on ChatGPT and asked, can you provide a summary of AI advances, across the research spectrum in oncology? And there's been lots of progress in the application of AI in oncology. There are many, many examples that you have seen on diagnostic imaging, digital pathology, which will be our focus today, personalized treatment planning, drug development, discovery, screening. And also there's now, I think the field is realizing that some of these, these things need to go to clinic. So implementation in clinic, regulatory aspects, and ethical considerations become important. So the question is that how can we actually unlock the transformative potential of AI to basically revolutionize patient care? And within the context of pathology, okay, so we've been relying on glasses slides for many, many years. And so we have these archival material that if you get one of these, let's say, Perio GT450 scanners, it takes 450 slides at the time. And in a few hours, you can actually digitize these slides. And then that provides a basically massive amount of data that you can actually, that is suitable for machine learning models, and specifically deep learning models. So how can we actually benefit from these AI models within the context of pathology, and specifically, let's say, digitized pathology slides? So one application would be basically improving, clinical pathology efficiency by AI, triaging more complex and difficult cases for expert examination, additional molecular testing. So this could be sort of like computer-assisted diagnostic systems or diagnostic adjuncts to pathologists. The second thing that we can actually use, and it's very exciting, is predicting, outcome directly from from these images, and also trying to link them to molecular subtypes with the hope that hopefully you can actually maybe eliminate some of the need to perform genomic or molecular testing, or, even like further refine the molecular subtypes. And from the basic science perspective, data integration to generate basically biological insights, like, from spatial omics, and linking them to bulk and single cell sequencing data becomes very interesting. So these are basically the focus of my lab. And then, so what we try to do is we take pathology images plus, omics data, and then with the hope to generate clinical and biological insights. The focus of the lab is mainly translational aspects as well. So for this to happen, so the vision of the lab called AI Medicine AIM Lab, so it's basically to build this, what we call plug and play AI engine that would take in various data types, and then we will try to generate basically clinical insights. So we started into October 2019 with one cancer, ovarian cancer. And then later on, we added prostate, endometrial cancers, lung cancer, and so on and so forth. We are at the point that we have active projects in 12 cancers. And I'm not listing the projects that we are discussing with colleagues as well, but these are active projects in the lab that I'm happy to say that, we have more than 10 various projects on more than 10 cancers. And the whole vision of the lab, again, is to build this AI, plug and play AI engine so that we can actually scale and have an impact in the department and in digital pathology AI field as well. So today, what I'm going to talk about is mainly, the second item. So trying to predict outcome from histopathology images. So we have active projects on endometrial cancer, prostate, low-grade gliomas, ovarian cancer, bladder cancer, specifically, with this focus. But I'm going to focus on endometrial cancer, which is, the paper that actually got accepted just this morning in Nature Communications, and excited to share the details of this study with you. And later on, I will talk about some of the things that we are actually doing at the moment in the lab. So a little bit of background. So molecular subtyping of endometrial cancer was actually done with the TCGA team in 2013, that they came up with basically 4 molecular subtypes, and basically defining endometrial cancers, or classifying them into cases that are called ultra-mutated cases with polymutations, and microsatellite instability, cases that have copy number, what we call copy number low, and also copy number high cases. And then these 4 molecular subtypes, they basically have correlation with the outcome of patients. So one thing that, is important to mention with TCGA studies, all are based on either whole genome sequencing or transcriptome sequencing, and they're all on frozen samples, So the question is, even if there is a clinical implication or clinical use for these studies, implementation in the clinic becomes problematic because these are all on frozen samples. So a team led by Jessica McAlpine and also David Huntsman and also Dr. Blake Jilks, they came up with this idea, with this bioassay. It's called Proactive Molecular Risk Classifier for Endometrial Cancer, or PROMIS. And it's basically a clinically applicable approach to identify the 4 molecular subtypes of endometrial cancer from formalin-fixed paraffin-embedded material. And this was done concurrently with another team from Netherlands, Transportec, that they basically came up with the same findings as well. And then this assay has been validated and in various rounds as well. So having this PROMIS classifier, so on the left, what I'm showing is, the molecular subtypes alpha of TCGA, and on the right, basically, these are the subtypes that, is provided by PROMIS, basically mimicking what we actually, mimicking the TCGA molecular subtypes, including polymutant cases with MMRD, and the difference was that, copy number high cases are the ones that usually have P50 abnormality measured by immunohistochemistry. And another group patients what we call NSMP, which is defined by exclusion, meaning that if it is none of these three, it becomes NSMP or no specific molecular profile. The interesting thing is that, so obviously, you know, cases with P53 abnormality, they do really bad and compared to the rest of the patients. And the patients with basically NSMP subtype, they're defined obviously, first of all, by exclusion. They represent 50% of cases, but their outcome is heterogeneous. So, and then, so the PROMIS and TransPortec, you know, the Dutch basically classifier, been adopted by WHO guidelines in 2020. And now it's part of ESCO classification, risk stratification, and also FIGO staging as well for endometrial cancer. Now, so we asked this question, so when we started this project, so okay, having, let's say, digital slides associated with these cases, can we actually try to refine these molecular subtypes? So this was a work done by 3 members of my lab, and Amir Dabansari, Hossein Farahani, and Mariam Asadi from my lab, that they did this work. Obviously, huge collaboration and you know, support from the OFCARE team, including Jessica Calpine, David Huntsman, and also Blake Schiltz, and made this happen. Without them, it would have not possible to do this. So we asked this question of like, can we actually build a classifier and a deep learning based classifier that would take again these images and would classify P53, differentiate P53 abnormal cases from NSMP cases. So just a little bit of a background on how these models work. You take basically these scan the images. These are hysterectomy slide images. We do a bunch of quality checking, identify tumor regions with deep learning models. We chop these into small pieces, and eventually we try to, you know, this is the deep learning part that we eventually come up with the subtype of a patient. So our discovery set was the Cancer Genome Atlas project, TCGA, and also data from Germany as well that was used to develop, basically to validate the PROMIS classifier. That was our discovery set, and our held out set was our local BC cohort. So what I'm showing here is the results of classification, differentiation between P53 abnormal cases and NSMP cases, just off of the slides. And what I want to draw your attention is over here, like the balanced accuracy is around like 83% for discovery set and 80% for the, let's say, held out or confirmation set. And so it's not bad, but it's not great. So then we ask this question of like, okay, so what the classifier is basically doing is that he's taking, let's say, NSMP cases. In roughly 80% of cases, it does a good job of classifying those correctly that were defined by molecular assay into NSMP, and roughly 20% as P53 abnormal. So you can consider that 20% as error, but you can actually look at it in a different way, which is like, okay, so 20% of NSMP cases are roughly, they look more similar from the histology perspective to P53 abnormal cases. So, and that was the question that, you know, came to our mind, that yes, I mean, these look more similar to P53 abnormal from the histopathology perspective. And now the question is that do they actually, and we know that P53 abnormal cases, they do worse compared to the rest of the patients. We asked this question, actually, are these patients that are NSMP by molecular, but classified as P53 abnormal by AI, do they have actually bad outcome compared to the rest of the patients? So, and we were actually, it was very interesting to find out that these patients that are NSMP but classified as P53 abnormal, they do actually worse compared to the rest of the patients, and it was confirmed in our confirmation or held out set. This is the progression-free survival curves that I'm showing. And on the disease-specific survival, again, we see the same trend that these patients do worse compared to the rest of the patients. And this is actually independent of its stage and grade for these patients. We took these cases and basically, we thought, okay, maybe with it sequencing, we sequence a few genes to see, if there is any mutation that would define this subtype. And the only thing that came up was p53 mutations. And we saw that over here. So these are the p50, what we call p50 abnormal like NSMPs, that they were enriched with p53 mutations. So these are the cases that are negative for p53 staining by IHC, but they have p53 mutations. So our classifier is actually picking up some of these cases, but there are some cases that are not actually, do not have p53 mutations either. So we said, okay, how about we just remove all the p53 mutant cases from this subtype? Does this biomarker survive if we actually remove those? And you're happy to see that actually those, even if we remove p53 mutant cases, we still see poor survival compared to the rest of the patients. We also took these cases and then did shallow whole genome sequencing. And what we observed is that, the patients that were classified as p50 abnormal like NSMP, they had higher copy number burden. So I'm showing, this is called copy number frequency. These are amplifications, these are deletions. This is like an NSMP case. You see like a lot of basically amplifications and deletions in p50 abnormal like NSMPs, but not in NSMP. And if you look at the discovery set and also the TCGA set, we see the same trend that p53 abnormal like NSMPs, they actually have higher copy number burden compared to the rest of the NSMP cases, but still less than p53 abnormal cases. So insights from expression profiling. So we looked at the expression data for these cases. We just did basically simple clustering to see if these patients cluster together. And we realized that, again, so over here, blue represents p53 abnormal, NSMP red, and green is p53 abnormal like NSMPs, that we see that they're evenly distributed across basically the two groups, meaning that expression-wise, they're not, there's no specific signature if you look at it in an unsupervised way, basically clustering. But if we do basically differential expression analysis of these cases, taking NSMP as our reference and do differential expression analysis between p50 abnormal like NSMP versus NSMP, and also another differential expression between p50 abnormal and NSMP, we actually see upregulation of PI3 kinase, AKT, Wnt, and carrier signaling pathways, both in the p50 abnormal like NSMPs and also p50 abnormal like groups. But the interesting thing is that the list, when you look at the list of differentially expressed genes, there is absolutely basically zero overlap between the genes that are up or down regulated in p50 abnormal cases and also p50 abnormal like NSMPs. What this tells us is that probably, these two are different, because one hypothesis was that maybe these are the cases that will become p53 abnormal at some point, but doesn't seem like that is the case because the pathways are the same, meaning that, okay, so they look probably the same. So we see it in the slides, but from the gene expression perspective, it seems like, different networks are active in these two subtypes of cancer. So far, what we have done is now we have shown that this biomarker that we have found, we are seeing it in two different cohorts. We're also seeing differences in copy number aberrations, showing that this could be real. But for AI biomarker validation, what we need to follow some of the guidelines. And so one recommendation is the Institute of Medicine guidelines, stepwise best practices that you would need to have discovery, validation, confirmation, and validation cohorts. So we had a discovery and confirmation cohort. So, okay, so if we want to go with the guidelines and recommended guidelines, and best practices, we need a validation cohort. And that validation includes two things. So one is biological validity, which is, does this subtype exist? So at that point, we're sort of confident that yes, because at least, from the genomic perspective, we see differences as well. And also from the analytical perspective that, can our model find these cases given the color variation across different hospitals? We know this is a big problem for AI in deep learning models, that when you even have a really good classifier that works on the Vancouver data, as an example, if you apply it to data from Toronto, it will probably break because of the differences in staining and so on and so forth. So we took, fortunately, we had access to a data set that Jessica McAlpine has been collecting. It's called Cross Canada. So this was from 26 hospitals across Canada, 416 cases that we had access to, and we had access to slides. And then this is the breakdown of the cases across different hospitals. And then what we saw is that this biomarker, was actually validated on the what we call Cross Canada cohort, that again, P53 abnormal like NSMPs, they have worse outcome compared to the rest of the patients. And this is having this in mind that the classifier was locked initially, and then we just applied that very same classifier with all the same parameters to these data sets without any changes or tweaking of the parameters. So what is the clinical implication? So ESCO and ECP 2021 risk group classification suggests basically including molecular subtypes, stage, histotype grade, lymphovascular invasion, and presence of residual disease. And you can classify patients in low, intermediate, high, and advanced metastatic risk groups. And that guides basically adjuvant therapy. So the question that we asked was, okay, does our classifier change actually the risk groups based on these guidelines? And the way we can do it is that all these cases that are classified as P53 abnormal like NSMP, what happens to the risk group if we consider these as P53 abnormal in the classification system? If we do that, we see that, in 19 of the 39 P53 abnormal like NSMP cases that we had access to other data for those cases, in 19 of those, the risk group actually changes. And we didn't see any change in the other 20 simply because they were already sort of like high risk already. So what this shows us that this actually has a potential clinical impact as well, and this is independent of like other clinical and pathological parameters. Now, the question is that, so everything that we did is on hysterectomy slide images, and we can have even more impact if we basically we show that, this could actually work on biopsy slides, because if that happens, then you can, in a community hospital, you can do a biopsy, you can run an AI classifier, and then see if, that can actually change the management of patients in terms of like, do they need, full surgical staging, or they can do a simple hysterectomy. So we did this validation also like on endometrial biopsies, 220 patients across Canada. This is part of the same cohort. And these are basically the results that we got as well in terms of PFS and DSS, that we see P53 abnormal like NSMPs, they have worse outcome compared to the rest of the patients. And these are different curves simply because we have different cutoffs to identify these cases, and this shows that the classifier is pretty robust in identifying these high-risk patients. So, so I think what I'm going to do, I'm going to actually skip this part because this is going to be another like maybe 4 or 5 minutes, and we're close to 1 p.m. But one thing I want to highlight is that what we are doing is we are actually building a platform that we call Atlas of Digital Pathology from archival material, that in an unsupervised way, we can actually, with these deep learning and AI models, we can actually build representations or features of the slide images. And then this platform can be used for many things or applications. And one is that if you have as an example, an image, so you can actually query this platform to see what is the closest image or patient to that sample. And then what this helps us is that it can be used as a search engine, what we call like, Google for pathology, that if you have a case, you can actually find similar cases. This can be used for N-of-1 studies or for rare cancers that you have like a few samples you want to see, within the cases that have been collected, let's say in BC, what are the closest cases, what is the outcome, and then there are so many things that we can do with this platform. And also you can use it to run clinical trials and also run AI on small cohorts as well. And as a matter of fact, so we've used this platform and we can actually identify the very same findings from for endometrial cancer, the P50 abnormal like NSMPs from using this platform as well. And finally, so this is a platform this is a technology platform, part of the same platform that is also, this is another paper on Nature Communications in Press, that is the basically engine behind this platform that eventually takes all these H&E images, identifies the cells, and then builds networks of cells, and then eventually you can actually link that to outcome like in high-grade serous and bladder cancer. So those are just examples, but everything is done in an unsupervised way without having labels or annotations for the data. So with that said, I'm going to stop. It's at 1 p.m. now, and then get to acknowledgements. It's been such a pleasure to work with a fantastic network of colleagues in pathology, and other hospital sites, and especially Dr. David Huntsman and also Blake Jelks as well, who's been very, very supportive to actually make these things, help me to make these things happen. But also appreciate all the other colleagues in pathology department who have been very supportive and working with us on various projects. And finally, my lab members who did the work, and just, presenting what they have done. So the real credit actually goes to them and also to my network of colleagues and collaborators. With that, so thank you so much for listening. I'm going to stop talking.
So just thank you so much, both of you. So before opening this up to questions, I just want to mention next week's lecture, which is been given by Dr. David Goldfarb, a microbiologist from BC Children's Hospital, and it has the enticing title of What's New in Number 2. And you can imagine what this is about. Maybe you want to eat your lunch while you're listening to it, or maybe you may want to have your lunch beforehand. So we look forward to that presentation. So now let's open up the floor for questions. And for those who have to leave immediately, we look forward to seeing you next week. Torsten.
Yeah, Ali, thanks. I really enjoyed that. That NSMP group, so your images can create a stratification like you showed. Can you obviate the need to do the POL or the MSI testing? Can you just replace all of that molecular testing with the image analysis so that we can just get a quick answer with the one test?
Yeah, that's a great question, Torsten. We are actually working on that. So our initial goal was to actually do that, but the problem with poly cases was that we started with the Vancouver cases. We had TMAs for most of those cases, but we know most of the information is actually in the whole section of slides. So that will be one of the next steps that we will actually do later on once we get the whole section of slides for these cases. And then that may actually help with poly and also identifying MSI or MMRD cases. But that's a great point.
Are there other questions? I do want to point out how rare it is for a AI type classifier to validate an external cohort to this sort of degree, and it really speaks to the quality of the sort of study design and the way Ali does his research. Sorry, Andy Wang.
Oh, thanks, David. Ali, I just wanted to ask you, is there a methodological way for the AI engine to output what the discriminating features are that it's keying on to make these distinctions, or are we just resigned to it's a black magic box and we just have to trust it?
Yeah, great question, Andy. So yes, it is possible to spit out what areas in this slide the model is actually picking up. What we call that attention models. So basically where the model is putting more attention in classification. So we usually take those and then, review them with our pathology colleagues to see if those make sense as well. So
have you tried that, or is that future effort, or do you have any ideas on that? So that, does it make sense?
Yes, absolutely. I mean, so we automatically generate those. It's in the form of like a heat map that is overlaid on top of the slides that you can actually see where, you know, the model is picking up. So that's part of the models that we do, because with these models, I talked about the success stories, but part of that is that actually once we do this, you know, then pathologist looks at them and say, okay, yes, it makes sense, but in some cases it may not make sense, meaning the model is maybe picking up something that is garbage or it doesn't mean anything. We'll have to go back, and it helps with troubleshooting as well. So that's why it's important to have it as part of the pipeline.
Or it can show you that it's keying on something we're not recognizing, right? You know, and
yeah, absolutely. So my point is that, yes, I mean, it can identify some of those. Obviously, the machines are more, I think, they're more objective. But the point is that it has to, if in my experience, so if a pathologist doesn't see anything from the images, that means, you know, the AI model is not picking up something meaningful. It will pick up, if it picks up something and then pathologist says, oh yes, now I see this, that this could correlate with, let's say, outcome, that becomes an interesting thing, because it's sort of like maybe a subtle changes or something that was overlooked, that now we can do these association studies in a more objective way going forward.
Okay. So we're a little bit after time. And so I want to point out there is an applicant for the BMLSC leadership position who is giving a presentation on protein structure and function right after this. And Lisa Scully had sent around an invitation on that. So you should have that available if anyone's still hungry to learn more over on this Friday. So thank you both of you for the great presentations and for the response to questions, both in the chat box and also presented. And we'll look forward to seeing many of you next week. And if any of you have ideas for presentations you'd like to see, or people who would like to present who haven't presented yet, for instance, Andy, I don't know if you presented yet at Rounds, so it'd be great to hear what's going on with Flow. And I think that's a volunteering, so nod. So this is good. We'll be in contact. We'd look forward to that. If you have ideas for other types of presentations as well, which could be provided, please let us know. So take care, everyone. Have a lovely weekend and stay warm.
Thank you.